Okay, hello everybody. So this is the podcast version of the molecular biology of the gene lecture, which is a lecture on the human genetics module, which is a level 1 module on a biomedical sciences course at the University of Bradford. So we're going to be talking about today broadly are different aspects really that come under the broad title of molecular biology of the gene. So we're going to be considering to start with the structure of a human gene. So one of the things I want you to keep clear in your mind throughout this lecture, and in fact throughout the module as a whole, is the fact that the structure of genes and of the genome in humans, in higher order organisms, differs greatly to the structure and organisation of genes in more simpler organisms such as bacteria. So you will be hearing a lot about bacterial genetics throughout your degree programme, but you need to make sure that you keep a clear distinction between those facts and what the situation is in more complex organisms such as mammalian cells and indeed our cells. So we're going to think about how we actually go about the business of turning a gene on and off, controlling gene expression, and then we're going to consider the so-called central dogma of molecular biology, and this is a concept that DNA makes RNA and then the RNA makes protein. And we're going to look at exactly how the cell goes about managing that process. We're going to stop and think a little bit about how our genome's organised. We're going to consider the question one gene, one protein, which for many years was believed to be how gene expression was controlled. They had a gene, a heritable unit, and that unit was responsible for production of a single protein. We now know that things aren't that simplistic. And then towards the end of the lecture, we're going to start linking gene expression to different human diseases, looking at how errors in gene expression can lead to illnesses. And then finally, we're going to think a little bit, some detail, about how we can actually manipulate genes, manipulate DNA in the laboratory, so that we can do experiments and molecular biology enable the expression artificially or in vitro in the laboratory of genes in a controlled setting. So I've put this up to reiterate the point I just made. Eukaryotic genes are very different in both structure and function to prokaryotic genes. Please emblazon that in your mind for the next 3 years. Otherwise you may end up getting confused whilst answering essay and exam questions and actually writing very accurate facts about prokaryotic genes which are unfortunately completely incorrect for eukaryotic genes. So with that aside, let's start by considering then the basic structure of what a human gene looks like. So this is a cartoon of a human gene, a very simplistic cartoon. It's fair to say that more or less all human genes possess a very similar set of simple characteristics. So here we have, if you like, the actual main body of the gene itself. What you will notice is there are two colours here depicting different sections of the gene. The blocks that are coloured blue refer to exonic sequence, and the boxes that are coloured red refer to intronic sequence. So hopefully you may be aware already from A-level studies that genes in humans contain exons and introns. If not, then I'll explain what these are now. But effectively, if you look at a gene as it exists in your genomic sequence, the genome within a cell in your body, the coding information of that gene is split across a fairly large area into exonic and intronic sequence. The exonic sequence, the exonic DNA sequence, that's the information which actually codes for a protein. So that's the information, if you like, that we want to extract and then use to make a protein in the cell. The intronic sequence is kind of separating all these different fragments of coding exons. And we'll talk more about what this intronic sequence might do later on. A second thing, really, that I want you to note on this diagram is this light blue region here, which is called the promoter. The promoter is a region of the gene which is responsible for the control of gene expression. So the promoter region, not surprisingly, promotes the expression of the gene. So it turns the gene on and off, if you like. There are also these two regions here marked by black lines, the transcriptional start site and the transcriptional stop site. Now the transcriptional start and stop site are different to the start and stop site of the actual coding region of the gene. So in other words, the region of the gene which contains sequence which is going to make protein. So when you actually transcribe a gene from the DNA template and make mRNA from it, in addition to the coding region of the gene and the intronic and exonic sequence therein, you also have a short region upstream and a short region downstream of that coding information called the 3' and 5' this end untranslated region. So in other words, that's a bit of DNA which has been copied into mRNA but isn't involved in the actual coding sequence of the actual gene itself. And we've already discussed exons and introns. So the final thing to consider is that each gene way upstream of the promoter, sometimes many thousands of nucleotides away, also possess these upstream regulatory regions. And these regions are thought to interact with the promoter to offer fine tuning to the gene expression control that's going on. So it's kind of a second layer of regulation that's present in order to control expression of the genes. So just to recap quickly, a gene in your body is made up of exons and introns. The exons contain the information which is going to make the protein. The introns were thought to contain just junk DNA, spacer DNA, but we now know that that's not the case, and we'll talk about that later on. Either side of the coding region of the gene, you have a 5' and 3' untranslated region, which is bookended by this transcriptional start site and transcriptional stop site. The expression of this gene is controlled by the promoter region, and the promoter region itself is similarly controlled. The activity of this promoter region is controlled by these upstream regulatory regions. And in a nutshell, on a basic level, that's what a gene looks like in our cells. So I said that the actual introns and exons span a large region of the genome, and this kind of exemplifies that point. So the other thing you need to realise is that of the gene unit, of the gene itself, the vast majority of the gene sequence is actually intronic in nature. So in humans, if you look for example here at the CFTR gene, which is a gene responsible for making the CFTR protein, which is the protein that's mutated and doesn't work properly in people with cystic fibrosis, what you'll notice here is this big long solid green bar represents the actual gene itself, introns and exons, as it appears in the human genome on chromosome 13. As you can see here on the top, this is the actual number of base pairs in that chromosome. Excuse me. And as you'll notice, if we look down here, you're starting at around 117,100 and it finishes at around 117,300 nucleotides. So you're looking at around 200,000 nucleotides there. Of those nucleotides, the vast majority are actually represented by intronic sequence. So if you look at the thing below this solid green line here, what you'll see is a series of thin green lines, and each of these thin green lines actually represents an exon within the CFTR gene. So as you can see, the exon sequences are very small, just in some cases just a couple of hundred nucleotides, and then following an exon you will have a huge intronic sequence, sometimes many thousands of nucleotides long, before you hit the next exonic sequence. So the other thing to realise is that human genes occupy large spaces on chromosomes a lot of the time, but the majority of that space is occupied in fact by the non-coding intronic sequence. The information that's going to be used to make the protein, the information in the exons, represents only a small percentage of the total size of the gene. So how do you turn a gene on then? But before we get into that, I just want you to have a clear concept in your mind with regards to gene expression. This is very important. You need to realise that not all genes are expressed all of the time in all of the cells in your body. So this is a different way of talking about a concept called transcriptional programming. So this is the idea that, say, a retinal cell, so a cell of the eye, will have a different set of genes being expressed at any one time compared to, say, a liver cell or a skin cell or a brain cell. Yeah. So the transcriptional profile, in other words, the genes which are currently being expressed in a cell, have a direct impact on, well, obviously, one, what that cell is doing, but two, what that cell actually is. So different cells have different types of transcriptional programming going on, and it's the gene expression control, the different types of genes being switched on and being switched off in cells, which effectively lends the cells their individuality. So it's not the case that if you have 20,000 genes in the human genome, every cell's expressing all those 20,000 genes all of the time. Only a subset are expressed, and that depends on cell type and obviously situation. So by situation, one example would be external stimulus. So it's true that during the cell cycle, which we'll be talking about in the next lecture, genes are continually being switched on and switched off in order to regulate that process. Same in development. During embryogenesis, different genes are switched on for different stages of embryogenesis, of organ development, and then those genes are switched off. Now, if this process isn't tightly regulated, serious complications can arise in these molecular processes like the cell cycle, and also in, if you like, gross anatomical processes such as embryogenesis. So how does a cell keep control over which genes are being expressed? It comes back to this region here, like I mentioned on the last slide, the promoter region. This is a region of the gene which is responsible for turning the gene on and off in concert with these upstream regulatory regions. So the first thing to realise, and I'll tell you this now if you didn't realise it already, that gene expression in humans is a very complex process. So don't get frightened. I don't expect you to memorise this or learn any of this figure. I'm putting this up because this is something that I actually will be showing the third year students on Thursday, and the third year students will be expected to understand this and to be able to discuss this if they're asked about it in an exam. So one, that gives you a little bit of insight into how the degree programme is going to progress in complexity and difficulty over the 3 years. But two, at this point now, I'd like you to not think that these things are simplistic processes. It's a very elegant, complex process, control of gene expression, particularly in higher organisms such as humans. Yeah. So I won't talk you through this. You'll recognise a couple of regions. This light blue region here represents the promoter region. This multi-protein complex here is actually the enzyme responsible for making mRNA from DNA. And the rest of it really are these upstream regulatory elements that we talked about. So you can break this down and simplify it, but ultimately it's a complex process that involves many different proteins. Not something you need to worry about, thankfully, at the moment. Although obviously if you want to read about this, you can find articles in Nature or in some of the more advanced textbooks. What I want you guys to think about is more the concept of how the process is regulated rather than worrying about all the different individual proteins at this stage. So a few facts then to start with. Well, gene expression in humans is driven by an enzyme called RNA polymerase 2. That was that multi-enzyme complex I just pointed to on the last picture. So RNA polymerase 2 is a huge multi-protein complex which is responsible for reading the DNA template information, in other words the sequence information in your genes, and converting that information into mRNA. Yeah. Now before it can do that, RNA polymerase 2 needs to be activated. And by activated I mean it needs to be recruited to the beginning of a gene, and it needs to be told to start copying that gene information into mRNA. So what does that? Well, that's achieved via a family of proteins called transcription factors. And these proteins function by binding to the promoter region of a gene and then triggering the recruitment and activation of RNA polymerase 2, which then converts the gene into mRNA. So this entire process, as we've mentioned a couple of times now, this recruitment of transcription factors and subsequent activation of RNA polymerase 2 is again on another level regulated by these upstream enhancer sequences. So let's look at that in a cartoon. What we have here, transcription factors being recruited to the promoter region of a gene. This TATA, or so-called tartar box, is a core element within the promoter. Practically all promoters have this TATA sequence to them. And transcription factors are often recruited to this core region of the promoter. Following recruitment of the transcription factor, RNA polymerase 2 will then be recruited to the beginning of the gene. And the entire process can be positively and negatively regulated by so-called enhancer complexes, which are often many thousands of nucleotides away from the actual gene that's been switched on. The mediator complex is particularly important in this enhancement of gene expression. Once you get gene expression being switched on, mRNA polymerase 2 then flies across the gene, copying the DNA into mRNA. Gene expression is underway. So that's how you turn expression on. Obviously, as well as turning on genes, it's very important the cell can turn off genes. If you can't turn off expression of a gene, then in theory at least you're continually going to be making the protein which that gene encodes for, and in some cases you don't want to continually make the protein. It might just be energetically disadvantageous to be continually making the protein, or the protein might actually do damage if it's continually produced at the wrong time during the cell cycle or the wrong time during embryogenesis. So in order to turn off genes, we also have things which are called transcriptional repressors. So in eukaryotic cells at least, these transcriptional repressors tend to work by interfering with the transcriptional activator function of enhancing proteins or transcription factors. So a good real world, if you like, example of a transcriptional repressor is the Wilms tumour protein. So this is a protein that's present in the developing kidney. And during development of the kidney, what the Wilms tumour protein does is it comes along and it binds to the promoter region of a gene called EGR1. When the Wilms tumour protein binds to EGR1, which itself is an activator of transcription, it turns the expression of the EGR1 gene off. So let me just go over that again. EGR1 is a transcriptional activator, so this is a gene that's present in the developing kidney whose job it is to go around and activate the expression of a number of other genes. What the Wilms tumour protein does is it comes along, binds to the promoter region of EGR1 and turns that gene off. So what the cell's doing there is it's turning off the expression of EGR1, but by doing that it's actually going to turn off the expression of a whole range of different genes. So rather than going around trying to individually switch off, say, 30, 40 genes, by turning off the thing that actually activates those 30 or 40 genes, it can kind of get a lot more bang for its buck. Yeah, much more efficient way of doing things. Now, if you mutate the Wilms tumour protein gene, so in other words, if the Wilms tumour protein isn't working anymore, then the consequence of that, well, think what the consequence would be. You would no longer turn off EGR1. In fact, EGR1 expression would just continually be on. And whilst EGR1 is on, all the genes which EGR1 switches on are also on and being overexpressed. And the biological consequence of that for an embryo, for a developing human, is that the kidneys start to develop tumours early in life. You've got uncontrolled expression of a range of genes, and the result of that is tumorigenesis in the developing kidney. So in this respect, we can consider the Wilms tumour 1 gene to be a so-called tumour suppressor gene. Yeah? So the expression of Wilms tumour 1 turns off EGR1, which turns off a whole range of genes. If that ability is lost, EGR1 overexpression leads to the formation of tumours. So Wilms tumour protein is acting as a tumour suppressor gene because it's in its absence, tumours develop in the developing kidney. Tumour suppressor genes are a very important family of genes, and we'll be discussing these in more detail later on in the module. So I mentioned the central dogma of molecular biology during the introduction. So the central dogma of molecular biology is that DNA makes RNA and RNA makes protein. So we showed this cartoon already. Transcription factors bind to the promoter region of a gene, recruit RNA polymerase 2. Positive enhancement of the entire process is given via these upstream enhancer complexes, of which the mediator complex is particularly important in humans, and this promotes the activation of RNA polymerase 2 and the conversion of DNA into mRNA. So this is another cartoon showing the same thing in a little bit more detail. So once you actually switch on RNA polymerase 2, what it does is it starts synthesizing in a 5' to 3' manner an mRNA copy of the DNA sequence. So we won't go into the specifics of what it needs to do in order to achieve that at this point, But you might want to think about a couple of things. Just, try and get your head around the mechanics of this process. It's copying information from a double-stranded piece of DNA. It needs to use one of the two strands as a template, so it's going to have to melt those two strands apart to get access to one of the two strands, from which it can then copy the information. As it copies the information, it kind of ejects out of itself an mRNA synthesized version of the DNA sequence. And this is what you're seeing in blue here. And that continues until the RNA polymerase II reaches the termination sequence in the DNA, transcriptional stop site that we mentioned earlier on. At that point, this newly synthesized mRNA is released, and it can go on to become protein in the cytoplasm. Now, we've already discussed at some length the concept that the genes contain exons and introns. It's important you realise that the exons and introns are actually both transcribed during gene expression. And it's only after transcription has stopped, only after RNA polymerase II has copied the DNA sequence into mRNA, that the non-coding sequence is removed from the mRNA prior to protein translation occurring. So this is a cartoon, if you like, of a newly transcribed mRNA. The coloured blocks here represent exonic sequence, and the black lines are intronic sequence. Once you've transcribed a new mRNA like this, it's called a pre-mRNA, a precursor mRNA, because it's not yet mature. We haven't finished the process of transcription fully because we've not got rid of the non-coding regions from this RNA. To do that, we need to initiate a process which is known as splicing. And what happens is another very complex multi-protein machine called a spliceosome comes along and recognises these intronic sequences on this pre-mRNA and moves along the mRNA, chopping these intronic sequences out, which then facilitates the joining together and production of what's called a mature mRNA. So now we've got an mRNA molecule that's much shorter. Remember how much of the actual gene was composed of the intronic sequence in that CFTR diagram from earlier on. 99% of the actual nucleotides are intronic. All of those get removed and degraded. And all we're left with is the coding exonic sequence, and that's what we're seeing here. So this will contain all the information required in order to make a protein. In other words, this contains a triplet DNA code, the codons, which will code for individual amino acids that when translated together produce a polypeptide or a protein. So how does that process occur? How does RNA make protein? Well, the first thing to think about, and you should have covered this in your tutorial already on comparison of prokaryotic versus eukaryotic cells, is that eukaryotic cells, mammalian cells, face another issue as well once they've actually produced a mature mRNA. And that issue is that gene expression occurs in the nucleus of a cell. Yet the translation of proteins by ribosomes occurs in the cytoplasm of a cell. So there's a spatial differentiation between the site of gene expression and the site of protein translation. Now what that means is that the cells have got another job on their hands before they can actually make the protein, and that job is to transport the mRNA molecule from the nucleus into the cytoplasm of the cell. Once an mRNA is exported into the cytoplasm, it can then associate with ribosomes, which go about the business of translating the mRNA sequence into a protein sequence. I don't want to go into a lot of detail about protein translation. You cover this in other lectures, and the actual process of translation by ribosomes in bacteria is very similar to this process which occurs in humans. So I'm not going to go into the details of how ribosomes work, other than to give you a couple of little conceptual things to think about. And that is that, for a starter, don't think about one ribosome coming along and making protein from one mRNA. So in other words, you haven't got a length of RNA, one ribosome comes along, spends 20 minutes copying the sequence, then falls off. The actual situation is more complex than that. Each mRNA is decorated by multiple ribosomes. And in this way, you can have multiple proteins being produced at the same time as ribosomes kind of queue up like cars on a motorway to move along the mRNA sequence. The other thing to consider is that mRNA doesn't stay around forever in the cytoplasm. RNA, unlike DNA, is a very unstable molecule, and it has a very, relatively at least, short half-life. The half-life of mRNAs differ, but none of them are around for a very long time. The most is a day or two, some of them for a matter of minutes. The control of mRNA half-life is another way of controlling, if you like, gene expression. Yeah? If an mRNA, once it's exported, is only around for 10 minutes before it's degraded and chewed up, then that only gives a cell 10 minutes to make protein from it. Yeah. If an RNA is around for 2 days after it's exported, then many ribosomes can bind to that RNA, use the sequence therein to make new protein. So why is that important? Well, think about what the cell might need to do. What if the cell needs to stop making a protein very quickly? It needs to stop immediately and no more protein be produced. In fact, if there's any more proteins produced, it could be very damaging to the cell. If that's the situation, you don't want to be stuck with loads of mRNA in the cytoplasm, which is very stable and which will continue to act as a template for the ribosomes for the next 24, 48 hours. Because even if you turn off gene expression by using the transcriptional repressor systems we talked about at the promoter region of a gene, you may turn off the actual conversion of DNA to RNA that's going on in the nucleus. You may turn off gene expression there. But because you've got all these stable mRNAs in the cytoplasm, the actual production of protein can continue for a long time after you switch off the actual gene that's responsible for making that protein. So mRNA half-life is another very important aspect of gene expression control. So one gene, one protein. So this was a theory that was put forward by Beadle and Tatum in 1941. For many years people thought it was right, up until, I guess, the early 90s, late 80s, so we're not talking too long ago. This stuck around for a long time. But we started to get a little twitchy about this hypothesis, really, as we started to understand more about genomic organisation. So, for example, we've now sequenced the entire genome of a model organism called C. elegans. C. elegans is a nematode worm. It's kind of been generous to make out that it's any way as interesting as this little fella here with his glasses, etc. C. elegans is a microscopic nematode, so it's you know, less than a millimetre in length. You need a microscope to really look at it, and it's comprised of just a few hundred cells. So a very simplistic organism, but a very useful model organism. We'll talk more about model organisms next year in molecular genetics. Anyway, this very simplistic worm that's less than a millimetre long, its entire genome contains 17,800 genes. So that's how many genes you need to make C. elegans, this simplistic worm. Now, that's all well and good, but we started to get a little bit confused when it turned out that Homo sapiens, i.e. us, well, our genome contains around 20,000 genes. So people started to scratch their head and really start asking the question of how on earth can more or less the same number of genes give rise to an organism as complex as humans, when a similar number is required to make an organism as relatively simplistic as C. elegans. It just didn't seem to make any sense. In fact, people used to think that we'd have around 100,000 genes in our genome. They just kind of plucked that number out of thin air based on, I guess, ego as much as anything else. Turns out it's wrong. We've got 20,000, very similar to this nematode worm. So how does that work? How are we able to create something so much more complex with no more genes than the worms? Well, it's down to this concept of one gene, one protein, and the fact that it's actually wrong. So it turns out that the junk sequence, the intronic sequence of genes, which people for so long really just kind of dismissed as not interesting. You know, this is the information that during splicing is removed and degraded prior to an mRNA being stitched together to make the mature mRNA molecule that the ribosomes then bind to and protein is made from. Now we now know that this so-called junk DNA is anything but that. In fact, a few months ago, there were several papers released simultaneously across Nature and Science, the biggest two research scientific journals in the world, detailing the completion of the so-called ENCODE project. This also got national news coverage on the BBC, etc. So the ENCODE project is perhaps the biggest leap forward in our understanding of genomics since the release of the Human Genome Project in 1996. In 2002. And what the ENCODE project looked at really was what on earth this junk DNA is doing. So it didn't just focus on the coding sequence, it focused on everything in the genome, including the intronic sequence, to try and work out what it was there for, what its job was in the cell. And they found lots of very interesting things. So the first thing they found relates to this one protein, one gene concept. So this is it in cartoon form. The idea was you had gene X and gene X made protein X and that was it. It's not the case. We now know that gene X can make protein XA, protein XB, protein XC, so on and so forth. In fact, some genes, there are extreme examples of genes where one gene can make upwards of 44,000 different proteins. So how can one gene encode for so many different variations of a protein? Well, it comes down to the fact that in higher organisms, we're able to manipulate how the process of splicing occurs. So rather than every single intronic sequence being removed during splicing and all of those exons being stitched back together, what you actually get is variations in the splicing process. Occasionally an intron may be left in. The exons might be shuffled around and stitched together in different ways. And all of those different processes allow you to generate different, subtly different proteins based on just a sequence that's present in one gene. So rather than in humans it being a case of 20,000 genes making 20,000 proteins, for us it's 20,000 genes making upwards of maybe 500,000 proteins. And now we can start to see where the complexity, molecular complexity, is derived from that allows an organism as complex as humans to be produced from such a small number of gene units. This makes sense as well because if you look at some more simplistic organisms, as you go down the tree of complexity, if you like, this alternative splicing mechanism, this ability to shuffle the information within a gene, becomes less and less prevalent. It's absent more or less from very basic eukaryotic cells like yeast, and it doesn't occur at all, or extremely rarely in prokaryotic situations where the intronic sequence doesn't exist. Now, obviously, if you start increasing the complexity of a system, you're more likely that there's more likelihood that things may go wrong, and this is one of the reasons why there's so many issues about understanding genetic disease. And in fact, this alternative splicing mechanism is often a cause of disease in humans, as we'll talk about later on in the module. So you can think of it like this, really, and people have proposed actually that the junk DNA, the intronic sequence and other junk DNA within the genome, is actually an indicator for organism complexity. So if you look at humans, I mean, you could argue that this is not a very complex organism, I guess, here. But if you look at humans as the most complex organism on the planet, which we are, we have actually got the greatest amount of so-called junk DNA within our genomes. And then as you go down towards very simplistic organisms like bacteria, there's very little junk DNA present. More or less everything in the genome of E. coli, for example, does something. It's of use to the organism. There's very little waste in there with regards to coding units. Doesn't have these big swathes of apparent non-coding DNA. So what is this junk DNA doing then? Well, we're not going to get into the specifics of it, but just to mention a couple of things. I've already mentioned the process of alternative splicing, and the intronic sequence is key at regulating that process. But there are also elements within the junk DNA itself which are capable of directly affecting gene expression and controlling gene expression in higher order organisms such as humans. And the best example of that, or the most famous example of that, are these so-called microRNAs. So these are very short nucleic acids which can interfere with the process of protein translation in a very subtle way to gently and subtly manipulate the transcriptional gene expression profile of cells. So it's all about layers and layers and layers of regulation. If you've got something very simple, then it doesn't need a lot of regulation to make it work. If you've got something very complex like a human, then you need to have very complex regulatory systems in place in order to make that complex organism work. Yeah. We'll go into microRNAs and other non-coding RNAs and in a lot more detail in year 2 and then year 3. So we've talked a little bit about how gene expression is controlled, but how do errors in this process, how do errors in the molecular biology of gene expression lead to actual disease? We've considered transcription. There are lots of examples of errors in transcription factors that lead to disease, but perhaps the most commonly quoted one is overexpression of certain transcription factors. A good example being the transcription factor MYC, which is known to be a prevalent occurrence in numerous cancers. So this is not massively different to the Wilms tumour example I gave earlier on. If you have a protein whose job it is to control the expression of other proteins, and this transcriptional control factor is itself overexpressed, then you're no longer dealing with just the overexpression of one thing. As a knock-on effect, you're going to get overexpression of anything that this transcription factor itself regulates. So when you start messing around with the expression and amounts of transcription factors in cells, it often leads to disease, and often that disease can be cancer if a consequence of uncontrolled gene expression is accelerated cell growth, for example. It can also be that if just one copy of a gene's mutated, it can lead to a problem with regards to disease as well. So there's a term called haploinsufficiency. So you should be aware that you get one chromosome from your mum, one chromosome from your dad. So in other words, you've got two copies of all the genes that, in your cells, and in most cases actually you can lose one copy of that gene, and it won't have any direct effect on you with regards to illness. You'll be asymptomatic. That's because there's another working copy which can kind of facilitate, gene function to proceed even in the absence of its sister copy. It's like if you're on an aeroplane and one of the engines go out, you don't crash, you've still got one working engine and it can get you home. Same principle in genetics for most of the time. However, in this situation of haploinsufficiency, that's not the case. This is a situation where if you lose one copy of a gene, the other copy that's left isn't actually capable of maintaining function. An example of where this occurs is with the DNA damage protein P53. So you'll hear a lot about P53 over the degree. It's a very important protein. It's mutated in over half of all cancers, P53, and it's responsible for monitoring DNA damage events and then preventing cells from dividing if they have damage to their DNA. We'll talk more about it during the cell cycle lecture in the next lecture session. You can also get dominant mutations. So this is where you have a mutation in one copy of the gene, the other one's fine, but even though you've got a working copy, you still end up having the disease. And that's because the mutation, the dominant mutation, causes what's called a change of function effect to the protein. So we're now no longer dealing with the loss of a working copy of a gene. We're dealing with the gain of a different function in one of those genes that's causing disease. So the fact you've got some working copy left over on the other chromosome doesn't account for anything, because the actual disease is being caused by a change to the way that the mutated protein is working in the cell. A good example of that is this TFCP2L3 gene, bit of a mouthful, which is prone to dominant mutation, and this dominant mutation leads to a disease called genetic deafness. You should be covering more about, you should have covered already about inheritance of recessive and dominant Mendelian genetics, etc. from Dr. Porter, and probably at A level. So you've got the genetics background to dominant diseases. This is the molecular reason why those kind of mutations lead to disease. So we can also get disease from aberrant processing of mRNA. So we've already discussed how alternative splicing, how mixing up the information within a gene in different ways can allow one gene to make many different types of proteins. And that's a really important key process in gene expression, and it allows us to be as complex as we are as an organism. However, occasionally it can go wrong, and when it goes wrong, this alternative splicing, it too can lead to disease, obviously. So we can use a Wilms tumour gene again here as for an example. Wilms tumour, the Wilms tumour gene itself actually undergoes alternative splicing following transcription. So in other words, you transcribe the Wilms tumour gene DNA into an mRNA, and the mRNA can then be spliced in different ways to produce different proteins. Now in the normal situation, what actually happens during splicing is that the alternative splicing mechanism produces two variations of the Wilms tumour protein that are in equal balance with each other. So every time you express one copy of the Wilms tumour 1 gene, you get these two variant proteins being produced in equal amounts. And it's the equal amount of these two proteins which is key for allowing them to function correctly. Now what can happen is, if there's an error in the alternative splicing mechanism, that results in a reduced amount of one variant of the mRNA and an increased amount of the other one. So rather than alternative splicing producing two different protein variants in equal amounts, one of the two protein variants is preferentially produced at the expense of the other. And then we have an imbalance in these two different protein isoforms, and that imbalance leads to disease. Similarly, errors in mRNA translation can lead to disease also. For example, mutations in a translation initiation factor. So this is a protein required for the initiation of translation. In other words, it kickstarts ribosomes along mRNA to make protein. This EIF2, eukaryotic initiation factor 2. If you have mutations in this gene, you will go on to develop a neurodegenerative disease called VWM, vanishing white matter. Similarly, there are proteins responsible for releasing ribosomes from mRNA, eukaryotic release factor 3. Mutations in this protein have been associated with a particular type of gastric cancer. And in fact, there are mutations which affect tRNA transfer proteins, the ribosome itself, and a range of other components of the translation machinery. And in these cases, it can lead to a multitude of diseases such as anemia and, again, several neurodegenerative disorders. So to finish with, then, I'd just like to consider how we're able to work with and manipulate genes in a laboratory setting. So I mentioned the PCR reaction during the introductory lecture. The polymerase chain reactions was a really important discovery for molecular biology. In fact, it probably gave birth to the field of molecular biology to a large extent. It was invented by Kary Mullis in the mid-1980s. And basically, in a nutshell, it enables the amplification of a specific region of DNA from just a single molecule of starting material. So there are two key elements in that sentence. It's a really nice way of summing it up, that sentence. The two key elements are a specific region of DNA, and the second thing is from a single molecule of starting material. Now, why are those important? Well, it's pretty useless to just be able to make more DNA non-specifically. If you want to work with DNA in the lab, it's likely you want to work with a specific gene. So you need a way of specifically amplifying and synthesizing just that gene, just the DNA sequence you're interested in. Similarly, often we haven't got a lot of starting material from which to amplify the gene. This is particularly true in the case of forensics, obviously, crime scenes. Also, it can be true if you're working with patient samples which have been stored historically, where we may have very little of the original patient sample left. So PCR's key now in a range of different applications in biomedical science, such as diagnostics. We use PCR to diagnose bacterial infection, viral infection, in addition to a number of genetic disorders. Research-wise, it's key to gene expression profiling, so we need to use PCR to see how much a gene is expressed in a particular cell or organ site, for example. As we mentioned in the introductory talk, it allows us to do gene cloning, to artificially clone and express gene products for medical benefits. And also, legal disputes are often settled via PCR now. Paternity testing is actually carried out via PCR tests. So this is a little GIF image of a PCR process. You can leave this playing and watch it at your leisure, really, on the lecture slides. But what you need to do initially when you do PCR is you've got a double-stranded DNA template. That's your starting material. That might be some blood at a crime scene, or it might be a bit of a biopsy of a tumour or whatever. You have a starting material which has got double-stranded DNA in it. Before you can amplify that DNA, as you should know now from DNA replication lectures that you hopefully have in cell biology, you need to separate the double-stranded DNA. Now, in the case of PCR, we do that by just heating up the DNA in a tube. Heat it to 95 degrees, the double-stranded DNA will melt, exposing the two complementary strands. Once you've exposed the two complementary strands, the next thing you need is you need to anneal two primer sequences. So DNA replication, DNA synthesis always requires a primer template junction, this here, in order for the DNA polymerase to make new DNA. If you don't have a primer template junction, you can't do DNA synthesis. Now, the beauty of PCR is that we choose a primer which we know will anneal specifically to a chosen region of the DNA in our sample. In other words, we choose a primer which we know will anneal to the beginning of a gene of our choice. Once you have the primer annealed to the specific region of the gene, DNA polymerase comes along and starts copying that DNA sequence to create a newly synthesized strand of DNA, which is complementary to the template strand. Now, the clever thing about DNA polymerase in PCR is that Kary Mullis realised you could use DNA polymerase from isolated from an organism called Thermus aquaticus, which is a bacteria that lives in hot springs. So by using the DNA polymerase from a thermostable or thermotolerant bacteria, it enabled him to heat the DNA to 90 degrees in order to melt the strands, cool the DNA down to allow the primers to anneal to the template, and then elevate the temperature again to an optimal temperature for this TAC DNA polymerase, Thermus aquaticus DNA polymerase, to operate. And then once Thermus aquaticus DNA polymerase has copied a new DNA sequence from the template strand, you can heat the entire contents of the tube up again to 90 odd degrees, melting these two new strands and allowing another round of PCR to occur. So the fact that you're using a thermostable DNA polymerase means when you heat to 90 odd degrees, the enzyme isn't denatured. If you use the DNA polymerase from E. coli, which goes at 37 degrees, every time you elevated the temperature in the tube to 90 degrees, the DNA polymerase enzyme will be denatured and it will be useless. So you wouldn't need to, every time you did an extra cycle of PCR, you would need to add more enzyme to it. Time consuming and costly. By using TAC DNA polymerase, a thermostable DNA polymerase, that's not necessary. And you can automate and cycle the process many times before DNA polymerase from TAC works. So if we can synthesize DNA by PCR, how do we actually know what DNA we've made? How do you read the DNA sequence? So this is Sanger sequencing, which we mentioned briefly again in the introductory talk. The Sanger sequence basically made sequencing of DNA simple, fast and safe. And it was the Sanger sequence that was used to complete the Human Genome Project. However, these days, Sanger sequencing has been replaced by so-called next generation sequencing, which we'll be going over in more detail at level 3. For level 1, I'd like you to try and get a bit of an understanding of Sanger sequencing, so you need to do a little bit of extra reading from the core textbooks on how Sanger sequencing works. This is a diagram showing the principle of it here, and in a nutshell it works by synthesizing the template strand of DNA that you wish to sequence with a mixture of different nucleotides, each of which contains, at a low concentration, a nucleotide which will terminate the synthesis of a DNA strand. Now, I don't want to get into more detail than that, but you can look at the core textbooks and do a bit of additional reading to try and get your head around how Sanger sequencing works. Now, as we said in the introductory lecture, Sanger sequencing techniques took 13 years, billions of dollars to complete the human genome sequencing project. The next generation sequencing machines can sequence a human genome in a matter of hours for a fraction of the cost, for a few £1,000s. So just a few summary and take home points from this lecture then. So we've looked at gene structure and function, and hopefully you've seen that it's very different to the process that goes on in bacteria. We've talked about how gene expression is controlled by transcription factors, both transcriptional activators and transcriptional repressors. You should understand now that human genes contain coding and non-coding regions, and the non-coding regions are removed by splicing, and that alternative splicing and shuffling the information within a gene in different ways allows us to generate organism complexity. So in other words, if we weren't able to use the information within a gene in many different ways to produce many different proteins, we wouldn't be able to generate an organism as complex as humans. Other factors are also involved in organism complexity, such as microRNA, which are small regulatory elements which control or help control protein translation. And they're beyond the scope really of this at level 1, but you need to be aware that they exist. Don't worry too much about how they operate for the time being. We'll cover that in more detail in years 2 and 3. And as a growing number of human diseases have been linked to errors in the processes we've talked about, there's an even bigger drive to understand in more and more detail how these processes work. We've looked in quite, pretty broad level 1 detail today at gene expression control. I'll give you an insight into some of the more complex aspects, but the reality is, even at the cutting edge, we still don't fully understand how these processes work yet. We're still every day making new discoveries about how they're regulated. And we finished by just giving you a brief insight into PCR and Sanger sequencing. Sanger sequencing you need to do a little bit more reading about on your own. These are two core techniques which kind of underpin modern molecular biology of the gene. Right, so thank you for your attention. Hope this podcast proves useful during revision, and look out for more podcasts in the series. Thank you.